Oh, welcome back to those of you who are watching the series of pitches for Innovation Week 2022. Today, on day three, we're welcoming Dr. Scott Tenenbaum, who is from the SUNY Polytechnic Institute in Albany. And he's going to talk to us today about what he has been dedicating his life to for the last several years and what kind of type of help he's looking for from students like yourselves. So, everybody, all ears, and we'll pass it off to Scott.
Thank you, Kelly. So I am the head of nanobioscience at SUNY Poly, and I am also the CEO of this new spin-out company called SXRNA Technologies. And I'm hoping to tell you everything you ever wanted to know about SXRNA, but we're afraid to ask. I can advance. There we go. So most of you have heard of DNA and protein. RNA is the kissing cousin of DNA, and it along with DNA and protein represent the three major building blocks of life. So some of you hopefully will remember from your basic high school biology class that DNA makes this really cool helical structure called the double helix, which is really pretty boring. Unlike DNA, RNA folds up into all kinds of millions of different structures depicted here. It's typically single-stranded as opposed to double-stranded like DNA, and that's what allows it to fold up into all these amazing structures, and those structures do work inside the cell by pushing buttons and making things happen. And it's that structure in RNA that we are exploiting. So normally in life, RNA forms these structures intermolecularly, meaning that it's one piece of RNA that folds up into these structures, and that's depicted here in 2 dimensions. In this case, we're making this little cute stem loop. And so the sXRNA technology is focused on turning what was a single piece of RNA forming a structure into two pieces. That's our technology. So we've turned a one-piece structure into a two-piece structure. And in doing so, we've turned this into a switchable module. So a little bit more about the basics of sXRNA. So we synthesize or make one of the pieces of RNA in the lab. We call that the bait RNA. And we've designed that informatically to interact with a second piece of RNA that we can either put into a cell or put into a test tube. It depends on what the product is. But we've designed this first piece of RNA called the bait to interact with a second piece of RNA that we call a trigger, coming from like a pathogen like COVID or from a cancer cell. And when the two come together, they form the structure that we want. So we've made this switchable. This is the off form, this is the on form, and we can control whether these little RNA nano switches are on or off by requiring that both pieces be present. And typically, we're requiring that one of the two pieces is coming from a pathogen like a virus or from a disease cell. So the know-how or the technology behind how you design these two-piece switches have rules, and those rules are a relatively comprehensive basis for 3 patents that I've already issued, and our company, sXRNA Technologies, has the exclusive rights. We've licensed these from SUNY, and we have the exclusive rights to commercialize basically any design and commercialization of two-piece RNAs as a switch. So here's a cute little animation that I paid way too much to have made. But essentially, we have our bait RNA in its off form. It interacts with the trigger RNA, folds up into the conformation that we wanted, in this case, that stem loop. And here we're attracting an RNA binding protein or a protein that's going to interact with this. And that's how we do work. So when the structure forms, we can have something interact with it like a protein. So we can tell whether the structure formed or not, or that indirectly tells us whether this trigger RNA was present or not, based on whether the protein interacts with it. So we are in the process of trying to commercialize this in 3 main products. So the sXRNA technology, as I've described it and as it's been patented, is what we call a platform technology. It can be plugged into lots of different things. We're in the process of deploying it in 3 major areas: messenger RNA therapeutics, which most of you are pretty familiar with because it's the basis for the new vaccines for all the COVID vaccines, diagnostics, specifically point of care diagnostics, and in biomanufacturing, a new emerging area in the pharmaceutical industries. So as I mentioned, we have the exclusive licensed rights to the sXRNA intellectual property, of which there's 3 issued patents. There's several pending as well. And we have a team I'm the CEO and CSO. Ed McNamara is a local business person with lots of medical and health field experience. He's the COO. Mike Barnes is a former GE legal counsel. He's our legal counsel. And Dave Wojcicki is our CPA. So I'll briefly mention the three product areas in the time that I have and can go into more detail later. So messenger RNA therapeutics, as I mentioned, are huge right now because of COVID. Two of the main vaccines, Moderna and Pfizer, were both messenger RNA vaccines. It's a growing industry. One of the limitations with the current technology is that where instead of putting the protein in the person, which is the typical way you vaccinate, like a protein from a virus, instead we put the instructions for that protein, that's the messenger RNA, into the person, and their own cells then make the protein, and they make an immune response to that. So for vaccines, it doesn't matter what cells the RNA goes into, just that the protein is made. But if we wanted to be much more target specific with a therapeutic, like let's say we want to use this technology to target cancer cells, then we need our drug, our messenger RNA drug, to just go to cancer cells and not the other cells. So our technology has the ability to make a messenger RNA therapeutic tissue specific. So in other words, it could go into any cell and it doesn't do anything. It's off, but our sxRNA technology, which we've adapted to the messenger RNA therapeutic approach, when it's on, recruits a protein that turns on its production. So we only get the protein produced, let's say, in a cancer cell. And that's the basics of the therapeutic approach. So we don't necessarily make the messenger RNA therapeutic, we make it tissue specific. And in this case, we could make like a suicide or death gene, which would kill the cell. Okay, as a diagnostic, we take the whole technology and we just put it in a test tube. So in this case, and it also is a huge market. The point of care diagnostic market is what we're targeting. Most or maybe all of you are familiar with point of care diagnostics now. That's the standard at-home COVID antigen test. So that's a great example of a point of care test. You're doing it yourself. You don't have to go to a doctor's office to necessarily do it. They're quick and easy. They have some limitations, but it's a growing market. So here we've taken our sxRNA technology and we put a label on it, and it is in the off form because it's the piece of RNA we make, our bait RNA, and it hasn't interacted with anything yet. Now, in this case, let's say we target the same RNA that we were targeting before in our cancer cells that was turning on our therapeutic. We could target the same unique RNA that's only present in cancer cells. In this case, we're doing it in a test tube. The two pieces of RNA interact, they fold up properly, form a new structure, this piece here, that isn't the stem loop. It's this thing right here is what we want to form. And now we can capture that or detect it using the same protein that we were detecting before. But in this case, it's in the lab instead of in a person. And we're using a super sensitive detection technology called enhanced Raman spectroscopy. It's a cool nanotechnology, and it allows us to detect this signal, which is now bound to the protein, which is bound to the surface of this diagnostic, and we can detect individual molecules. So it's extremely sensitive and pretty rapid, like within 10 minutes. Lastly is biomanufacturing. So about half of the drugs on the market now are produced by this new technology called biomanufacturing. It's not actually that new. But traditional pharmaceuticals are produced, they're small molecules, and they're typically produced by chemistry. Biomanufacturing is different. We actually use cells. They can be human cells, they can be monkey cells, they can be bacterial cells. But we use a living cell to produce the drug. So it's not chemically synthesized, and it's particularly challenging. Although it's a huge market, a great analogy for the biomanufacturing process is microbreweries. So microbreweries, small beer manufacturer, it's batch produced. So there's these literally giant vats where they produce the beer. In an analogous manner, biomanufacturing, they grow the cells, bacteria or other types of cells, in these giant vats, which actually are responsible for producing the drug that's going to be made. And then they purify the drug and sell it. And so everything is produced in batch. And so there's a lot of issues and pain points in the manufacturing process. And so we've introduced our technology into this platform. We use it as a cute way to increase yield, increase production time, and decrease variability from batch to batch, all three of which are real pain points for the industry. And so that represents the third of the three strategies that we are developing in our commercialization approach. is to reduce to practice proof of concept prototypes for each of these three commercial potential products, and then work with an industry commercialization marketing partner to actually sell, produce, and sell the product, and with some type of royalty sharing agreement and some type of an R&D continued contract. So with that, I will conclude and take questions.
Scott, that was great.
How'd I do on time there? Was I even close to 5?
A little over. That's all right, though.
All
You
right.
gave us a good education and a baseline to start understanding the limitless possibilities of what you're working with. The three areas of focus that you had mentioned, they're all hot, hot areas.
They
So
are.
one of your challenges you were saying before is to identify which area, which direction to go in first, and that which direction is going to be the most sustainable and profitable with a business model attached to it.
Yeah, and I can expand on that. Like I said, these are all three not only decent sized markets, they're all reasonable size. They're all pretty hot and emerging and growing. We have struggled with bouncing around with whatever is the sexiest one at the moment and whatever we're getting a little traction on. And we've actually pushed all three product lines forward with prototype proof of concept. You know, the idea is to have something we can put in a potential partner's hand and say, okay, here, test it yourself, see what you think, and we thought that's the best way to try and get some type of a sublicense arrangement or contract in place. We could use a lot of help on trying to decide which of these three might be our best market. So in general, we can group these three areas into what I would say cost benefit. And so the therapeutic field, I think, is by far the sexiest and the one we'd most likely love to get in, but it's the heaviest lift. I mean, therapeutics are a nightmare, and for a small startup, that's a heavy lift, especially in the capital region. You know, if we were maybe in Boston or out on the West Coast, maybe it'd be a little different story. But to produce a therapeutic in this area is a pretty heavy lift. So I think that may not be the ideal starting point for us. The point of care is kind of in the middle. Lots of neat stuff, but it's a different type of lift. It's not really quite as heavy since you're not putting stuff into people, but it is regulated. What your false positive, false negative rate is a little different, and there are some regulatory components to it. But it's a great market, and it's not going into people. And then lastly, our biomanufacturing is what we're probably going to wind up deploying first because it is probably the lowest hanging fruit, although we're not committed to that. Um, in that there's a ton of need, and probably we could get our foot in the door the easiest. The barrier to entry here is that it's a field that is riddled with historical requirements. This is the way we do it, and we can't change that because our drug has been approved using the way we do it. And if we make any changes at all, we have to go through the entire regulatory process again. So we can't change anything. The only thing most biomanufacturers would be interested in is playing with our stuff for maybe a new drug, not something that's already on the market. So there's some issues with how you get into that field. But in general, I think we've been leaning towards biomanufacturing and actually making a little kit for molecular biologists as an easy entry. It's a lower market, and you know it's not going to produce billions of dollars and might not even produce millions, but it could be a little kit that we sell to researchers like at RPI. They pay 400 or 500 bucks that accelerates their own cloning procedures and at least get out of the gate, generate a little revenue that way.
Great. That's now you mentioned not only, well, you've been working on this for several years and
Yep.
you've been looking at the business side yourself, doing customer discovery in the iCore program. Who are the actual key stakeholders that you're trying to convince the decision makers here?
So it's a great question, and it's specifically what the iCore kind of helped us with. So in all three cases, the end user is not our customer, and you kind of figure that out in the iCore. A point of care, as an example, point of care test diagnostic. Well, the person doing the test is the end user, but that's not actually our customer. Our customer are the diagnostic manufacturers like Abbott, Pfizer, you know, the people that make these diagnostics, that's actually who our customer would be. So trying to figure out how our value proposition fits with their existing portfolio or product line and how we can adapt our stuff to what they're doing, their test kit, for instance. is kind of where we're at. And I can say the same thing for all the others. For biomanufacturing, yeah, a person takes the drug, but Moderna, we actually have a relationship with already. As an example, that's our customer: Moderna, Pfizer, BioNTech. Those are our actual customers. And the same in biomanufacturing. Regeneron, the local, which is a big time biomanufacturer, local company out in East Greenbush, is a great example of that's our actual customer. And we've reached out to them to figure out how we get in the door. So there aren't millions of customers. There's actually a handful of limited ones. There's some overlap. Like Pfizer has a foothold in a couple of the sectors, but they tend to be kind of unique for each of these product lines. So we doing customer discovery for one product won't necessarily get us access to the customer of a different product line. So it does kind of mean we have to decide what direction we're going in before we put too much energy into the customer discovery.
Right, right. You've cast that wide net, as we say, and you've identified the possibilities, but now we need to find out who has the hair on fire
Right.
issue, as
I'll add
previously said.
another little answer or addition to that answer, which is relevant to the ICOR program. So I did ICOR with Kelly, and so I'm giving some shameless shout outs to that. But what we developed there was the diagnostic pipeline, part of our diagnostic, and what we were specifically going after, it was a metastatic relapse diagnostic. And I'll explain what that means. But how we discovered that was in the process of doing our customer discover. We talked to CDPHP, one of the biggest healthcare insurance providers in the capital region, but they're unique. They're a capital region. That's what the C stands for in CDPHP, Capital District. And we met with their top people, including their researchers, and they said, we were trying to figure out, is cancer a good thing to go after? What do you need? And they said, what we really need is someone to be able to tell us when someone is having a relapse of cancer, meaning not the first initial exposure, like someone who has breast cancer. That's your first primary. But after that, like in year 2, 3, 4, 5, 6, when they have a relapse, that's called a metastatic relapse if it's the same cancer. What they said was by the time we identify that using something like a PET scan or a CAT scan, we are limited to maybe one or two drugs, and those cost us $1 million each, and they don't really work. So we have to go spend $1 million to treat someone, and the prognosis is pretty bad. So we're kind of wasting money. If you could just catch that a few months earlier, even 2 months, but if you could catch it 6 months, now we've got dozens of drugs that we could use, and those cost $10,000 each. So just being able to get access a little earlier gets them in the game. So our customer there is CDPHP. And they said they would potentially even fund the work, and that it hadn't occurred to us that they help deploy products in the space, because at the end of the day, they're the customer of Abbott. They actually, they're the ones who buy the tests from the manufacturers. And so that's our customer too. And so it was a really cool process of how we kind of figured out who our customer was. In this case was an insurance customer or insurance agent, that it would never have occurred to us that's who we got to kind of figure out how to shape this.
That's a great story. I love it. Yeah, you guys were fearless. You were knocking on lots of doors, and you got some great answers. So it was wonderful that you did cast that wide net. All right. Well, I think we're going to have success finding you a team, Scott, if not three teams. And,
I hope so.
but we will follow up and get those student names to you as they come in mid-October, and see if we can get something running in the spring.
Wonderful. Nice seeing you again, and best of luck with the program. And give my best to Brett.
Thank you, Scott.
Thank you. Bye bye.